Hello. Okay, we're gonna finish membrane potentials today, and we'll briefly start endocrine system. Remember, endocrine is not going to be on the first test; it will be on the second test. But you guys are doing it in lab this week, so at least I can give you a little bit of like background information. Then President's Day, you guys have off. No, not yay. We've got shit to do. And I'm not super stoked about presidents in the current state. And then next Wednesday, you guys have your test. Yeah? Sorry, feel like I never see you. What the hell? And then we'll start working on endocrine again after that. I will also post some stuff for you guys about endocrine after your test.
What is it?
Today at noon, I'm going to take a little break to feed myself and photocopy some stuff right after this class. If you guys are interested in, Joe Stosich and I are going to do a little workshop for you guys on action potentials. It's a game that makes you think a lot and figure out whether you know where the channels go and what they have to do with anything. So it's going to be at noon in the STEM Center, first floor. I don't remember. I think it's 101 something. You'll find me. I'll be there at noon. So if you don't come, that's totally fine. But I want to ask you to be generous if it helps you and do it with somebody else. Okay? So play with it with somebody else. I've never done it before, but it got really great. There was a paper published about it, and the students who did the game performed way better than on their assessment than the students who didn't. So we'll just give it a shot. We'll try it together, okay? Either way, Joe and I will also be there to answer questions for you guys, and you can have a little extra instructor time. Your quiz is due at the February 13th, which is tomorrow, 5 p.m. Look at number 9 again. You don't have to do it right now. Just look at number 9 later, because people didn't realize that they could choose 2 answers, so I changed it so there is an E that actually has 2 answers, because there are 2 correct answers for number 9. So you could have chosen 2, but people didn't realize, so it was confusing. Okay, so let us finish this set of notes today. So what we did last time was basically finished up with talking about action potentials and repolarization, right? Okay, but I want to, because it's been a minute and this is complicated, I want to do the same stuff that we did before, but this time I'm going to have you guys fill in blanks for me to see if you're getting comfortable with terminology. Okay, so we're going to tell the same story that we told before. We're going to use this as a guide to tell it. We're going to say, okay, starting the story that we had before, we are going to imagine the process of generating an action potential and then repolarizing a neuron, right? So we're going to do what we did before, where you say, okay, neuron has the neuron cell body with the dendrites right here, okay? And then right here by Dana is still going to be our axon hillock, okay? And then that's the whole axon, and axon terminals all the way down at that end. Cool? Okay, so questions for you. And the terminology is really, really important. So before I do anything to this neuron, anything extra, the neuron is sitting at blank membrane potential. Resting membrane potential, which is what? About -70, at least in neurons. Okay? So it's just sitting here minding its own damn business. And then what I do is I dump a dump truck full of ligand onto it. This is acetylcholine, a ligand that binds to ligand-gated sodium channels on the cell body and the dendrites. Yes? So it's going to get dumped on and bind to not 1, not 10, 500, right? Ligand-gated sodium channels. That is going to blank polarize the cell. Depolarize the cell. Exactly. What is happening right now on the cell is a blank membrane potential. I'm depolarizing the cell. Nope, a blank membrane potential. You told me resting before, and now I'm a graded membrane potential. A graded membrane potential. Good. Ligand-gated, graded membrane potential. Okay. If that graded membrane potential is relatively massive here, like maybe here it goes from -70 to -40, -40. then it might be big enough to get this side, which is further away, and of course you dissipate a little bit of the charge, right, to minus 55. Minus 55 is important because why?
Threshold.
Threshold. What happens at threshold?
It's enough action potential to
Enough,
send it to
enough of a graded potential to open a voltage-gated channel. Voltage-gated what kind of channel? Yes. So I'm going to open my first voltage-gated sodium channel. When I have opened a voltage-gated channel, I'm in a blank membrane potential. A blank potential. Action potential. Yes? Okay? Right? Resting, graded, action potential. Right? Action potential, therefore, only occurs in cells that have voltage-gated channels, and that is only in muscle and nervous tissue. Everybody with me? Okay, so now let's do what we did before. I'm going to open the first voltage-gated sodium channel, right? Which is going to further depolarize because, of course, sodium is going to move in. So that means if this one got to minus 55 and then I flooded myself with sodium, I'm going to probably make it so that this one gets to minus 55. Yes? Okay. And that one and that one and that one like dominoes. Flip the first one, they all go down, right? But the last thing that we were talking about is the fact that one action potential is all well and good, but you're going to need to be able to have them frequently in order for the tissue to be able to be useful to you, especially with nervous tissue. And we'll talk about that in more detail today. So what I'm going to have to do is I'm going to have to depolarize all the way to the end, send my signal. By the way, that's what causes neurotransmitters to be released, for those of you who care. But I need to be able to reuse it quickly. So what I'm going to have to do is I'm going to have to chase depolarization with repolarization. Okay. Can we all agree that if I just massively flooded myself with sodium to create an action potential, even if I dumped another dump truck of acetylcholine down there, I'm not going to get another action potential because all of the sodium is already inside the cell. I've screwed up my gradient, right? So I'm going to have to fix it before I can use the cell again. So let's talk about how to fix it. The way that you fix it is, after I open this one, it makes sure that the neighbor opens. Then behind, what I need to do is three things. What do I need to do? Close sodium, yes, then open potassium, right? And then I still have to deal with the fact that maybe I'm at minus 70, but all of my ions are in the wrong place. What do I have to do? Sodium potassium pump, right? Okay. Same thing here. Open this voltage-gated sodium channel, close that one, open potassium pump. Yes? Does that make sense? Okay. So let's revisit the video that we looked at last time to make sure that this makes sense to you. Okay, so this is the video that we looked at last time, and it doesn't show any of the ligand-gated channels or any of the leak channels because this is just on the axon, right? It's not even showing the sodium-potassium pump, although it's there. It's just focusing on the voltage-gated channels. So what I want to do is I want to say, okay, I'm going to hit this thing with a zap, and the zap represents threshold, right? Okay, go ahead. Oh, it's not up there? Oh, damn it. Thank you.
Make sure we're recording.
I did record it.
Oh, good.
Why is it not up there?
It's not Monday, so it should know.
Try that. There we go. Okay. Okay. Come back, you little bastard. Okay. All right. So you ready? So, zap. That's threshold. So what's going to happen now? Voltage-gated sodium channel is going to open at threshold. Okay. So where am I on this graph now?
Phase 1.
I'm in phase 1. Yes, because the voltage-gated sodium channels are going to open. Everybody with me? Okay. I'm in phase 1 on that graph. Now keep going, although go more quickly because it's boring. Okay. Now where am I on the graph? I opened potassium channels. I'm not in phase 2. I'm right here at the top, right? Okay. So what happens is I was really, really depolarizing, and then I would prefer that they close sodium before they open potassium, but that's just not the way it happens in all cells. So I'm going to open potassium, which will at least stop depolarization, right? Okay. And now tell me when I get to phase 2. You guys tell me. So now I'm at the top. When am I going to get to phase 2? Right there. Yes. Now it's only potassium that's moving. Yes, only potassium that's moving. Okay. And then what's going to happen is I am going to go all the way down phase 2, and it always slightly overshoots negative 70 just because its diffusion is really fast. And then it'll just come right back up. Go ahead.
So phase 2 doesn't happen until you've closed sodium?
Yes.
It's not when you open potassium?
No. If you open potassium but sodium was still open, you'd just kind of be battling against one another. You really haven't started moving down dramatically.
Okay.
Does that make sense? Okay, so everybody okay with that?
No.
Go ahead.
So phase 2 happens when you close potassium?
No. Should close sodium, open potassium.
Close sodium.
Yeah. So at the top, they both happen. Close sodium, open potassium. Yes? And the order doesn't actually, it's not consistent from cell to cell, right? I would prefer that they close sodium first, right? Because you got to put up one toy before you get out the other one. But it works either way, right? It's just ions, right? Okay. And then when you're in phase 2, you only have sodium open. So let's just think about this for just a second. Sorry, potassium, stop doing this to me. Okay, it's all your fault, people. So I want to talk about what voltage. I'm not going to make you memorize other voltages besides resting and threshold, okay? But I want you to think about what's logical for the other ones, okay? So what do you think is a logical voltage without looking? What do you think is a logical voltage for the potassium channels to open and or the sodium channels to close? Yes, someplace past 0 up toward 30, right? Okay. So it's definitely not going to be minus 70, minus 55. It's not going to be minus 90. That would make a damn bit of sense, right? So it's got to be up there somewhere. And by the way, it varies from cell to cell, but it's always kind of consistent in that direction, right? So if one of them closed before the other, if we close sodium first, and you do in some cells, then sodium might close at positive 20, right? And potassium might open at positive 30. Or it could be the other way around, right? Okay. Anywhere between 0 and those. Good? Okay. And then, of course, what's going to happen, and I won't make you memorize this one either, is that eventually I'm actually going to have to reset my sodium channels because sodium actually has 2 gates. The first one opens at minus 55, right? But then this one closes, we said at, I don't know, positive 20, something like that. Positive 20, positive 30. But then we're going to have another voltage that resets it all together, right? And the other voltage that resets it all together would be coming down that other side, okay? Now, that's not a memorization question. It's just like, how do I make one thing happen after the other, right? Make it the voltage that you hit second or the voltage that you hit third. But it's not memorization. Go ahead.
So this all happens through the sodium potassium pump?
No, these are not pumps.
So
these are voltage-gated ion channels.
So they're just completely different channels, but they move in
The pump is not a channel. The pump is pump. The pump is
moving in the same direction.
no. The pump is moving exactly the opposite directions. These are diffusion through a channel. The pump is generating this gradient so that they will move, right? And replacing it after they do move. But the pump is totally different than ion channels. Yes? Pumps move things against their concentration gradients. Ion channels move things down their concentration gradients. Go ahead, Maria.
So are these channels on the same place where these other ion channels are?
No.
Or is it in a different?
No. I mean, this is oversimplifying. There are exceptions to this, but just to get you something to think about. Typically speaking, again, let's do this one more time. What kinds of channels tend to be abundant? In addition to all the resting ones. Let's not focus on the resting ones for just a second. What kinds of channels tend to be abundant on the cell body and the dendrites of a neuron? Ligand-gated channels, yes? Okay, good. And then I don't have any voltage-gated channels until I get to the axon hillock, yeah? And then I've got them all the way down. And where are sodium-potassium pumps? Yes. Every damn place, right? They have to be. They were there to set up the gradient. They're here to fix the gradient. They're everywhere, right? It's part of the reason that this tissue is so energetically expensive. Because not only do you have to generate concentration and electrochemical gradients for ions, you have to fix them after you mess them up over and over again, right? So I'm going to have ligand-gated channels and sodium-potassium pumps here. I'm going to have voltage-gated channels and sodium-potassium pumps all along the axon. Good? Okay.
I have a question, sorry. So I'm confused. Ligand-gated versus graded.
Okay. You're looking at 2 different nouns. A ligand-gated channel produces a graded potential. A voltage-gated channel produces an action potential. The channel is a physical thing within the cell membrane. The potential is what you measure by sticking a voltmeter in it.
Okay.
So don't confuse gated and graded, right? Put the T in there. Don't be an American. Everything has a duh in the middle for us, right? Graded, gated. Good? Okay.
So wait, the
Questions?
ligand is the graded?
No, it's not the same as the graded. Have you guys been talking to each other and telling each other wrong? No. The ligand is, somebody make that statement for me. How is it, what's the relationship between a ligand-gated channel and a graded potential? Opening ligand-gated channels causes graded potentials. What is the relationship between a voltage-gated channel and action potential? Opening voltage-gated channels, specifically sodium, causes action potentials. Yes? Okay? So the potential is the measurement. The channel is the channel. Yes? Okay? Okay? Got it? Yes? Okay, I don't believe that you've got it, but I am going to move on. Go ahead.
It doesn't seem like there's anything graded about it, though. It seems
Oh, okay.
action potential is an all or nothing.
No, that's action potential. Graded can be strong or weak depending on how much ligand you have. That's different grades, yes? Okay?
Sure.
Yeah, so that's why it's called graded. So if I dump 5 molecules of acetylcholine and it opens sodium channels, I get a little bit of a depolarization, minus 70, minus 67. But if I dump a whole bunch, I'll get a bigger depolarization, minus 70 all the way up to 0 maybe, whatever, right? That makes sense. That's why it's graded, okay? So different grades. So again, to reinforce what he just said and also what you guys are trying to grasp here, and that is that graded potentials can be strong or weak, but action potentials, once you open the first voltage-gated channel, how many do you open? All of them. Yeah, they're all or nothing, which is exactly the topic we're about to cover. They are all or nothing. So there is no strong or weak action potential because in a cell, once you open the first voltage-gated channel, you open all of them. It doesn't mean it's exactly the same as a different kind of cell. It means per that cell, all of them or none of them. Okay, so let's get to that all or none thing in just a second. Okay, so let's talk about, before we get to the all or none, let us talk about refractory periods. So the refractory period, so listen to this, it can reinforce what you guys are trying to grasp. The refractory period is a period, and again, let's talk about neurons right now. We'll talk about muscle cells later. In a neuron, there is a period of time when I am having or just had an action potential in which I can't have another one. So here's a picture, here's what I want you to imagine. I just dumped a boatload of acetylcholine, right, onto these ligand-gated sodium channels on the cell body, and I have an action potential going, right? Okay. When that action potential is happening, I can't have another one even if I have more acetylcholine there. And the reason that I can't have another one is really that I'm busy with the first one. So let's look at this one more time. And I want you to think about, okay, do you see that right now, right here, okay, I'm still in the middle of the first action potential. So regardless of if I dumped more, I couldn't have a second action potential. Does that make sense? Okay, so for that matter, I'm still in the middle of it now because my sodium channels can't get more open, yes? But I'm also still in the middle of it now because even if you got them to open, this other gate is closed. So when am I out of what we call the absolute refractory period, in which it's at least theoretically possible when I can get another one? And that is then, when the sodium channels are reset. Then I can have, when I set up my dominoes again, I could, for instance, flick them, right? Does that make sense to you guys? So what's going to happen there is, if we go back to this figure for just a second, I'm going to be really busy. My sodium channels aren't going to be reset for most of this action potential. Really only at the end, when I haven't quite fixed it, but all my sodium channels are like back in the right position, could I have another one. So the refractory period, there are two different forms. There's the absolute refractory period. Absolute refractory period means I do not care how much stimulus, how much acetylcholine or whatever you add, you are not getting another one because your voltage-gated sodium channels haven't reset. Yes, you haven't put your dominoes back up, so you can't have another domino row, right? So either most of your voltage-gated channels are still open from the last time, or they're still inactivated, in which that bottom gate is closed from the last time. Now, it doesn't really matter what potassium channels are doing during that phase because it's not potassium channels that generate the action potential. So this is primarily related to sodium channels. So how long does that last? For most of the action potential, right? Most of the action potential is when you actually get that thing. So this is what we call the absolute refractory period. Do you know the term refractory period from human sexuality? Oh, you do? Good. You probably weren't educated in Utah if you know where that is, right? Okay. The refractory period in human sexuality is the period of time after a climax when a male cannot have another climax. And it doesn't apply to women. Yay, high five!
Yeah.
Almost makes up for always getting paid less. But yeah, there is no impediment to back-to-back climaxes, right? But in males, there is a period of time after climax in which they can't have another one. When you're 14, it might be 5 minutes long, right? When you're 72, it might be 5 days long. But the period of time in which I don't care what she looks like, he looks like, I don't care what your thing is, no way, no how, right? No way, okay? So that's the absolute no way refractory period. Has to do with the position of the voltage-gated sodium channels. They have not been repositioned so that they could respond. Okay, but there's another time in which the refractory period is still going on. It's not impossible to get to an action potential. It's just difficult to get to an action potential. And it's difficult because either you're working against the potassium channels because they're still open, or you're in that slight hyperpolarization period in which you're not sitting at minus 70, you're sitting at minus 75. So let's look at this. So with this picture, so what you're going to see, this is absolute refractory period. I'm still busy from the first one, right? Okay. But if I get the sodium channels reset to the original position, now I could theoretically get an action potential, but it's going to be harder to get to because potassium is working against me, right? So now where I am is I'm in what we call the relative refractory period, which is this period of time at the end in which one of two things is true. Either your potassium channels are still open, which makes it harder to depolarize, right? Or you are slightly hyperpolarized, and instead of sitting there right at this point at minus 70, you're sitting at minus 75, minus 80. Now, if your sodium channels are reset, you could still get an action potential, but it's going to take more to get there. Whereas I could have started this action potential with 100 molecules of acetylcholine. This one might take 180. I don't know, I'm just making stuff up, right? But it's the relative refractory period, right? It's when it's harder to get an action potential, but it's not impossible. And the relative refractory period, again, has to do primarily with potassium, yes? But the absolute refractory period has to do primarily with which channels? Voltage-gated sodium, yes. You have to get them back into their original state before they could respond to another action potential. Now, with refractory periods, the refractory periods vary from the different electrical tissues, but really the electrical tissue that you need to reset the fastest is nervous tissue. It has the shortest refractory period, and then cardiac muscle. Oh, wait, no, and then skeletal muscle and then cardiac muscle. Skeletal muscle is kind of an emergency response tissue. You need to be able to like make it work quickly to get out of the way of a bus. And then cardiac muscle, and then the slowest reset is in smooth muscle, which is like the sloth of the electrical tissues, right? Doesn't really do a hell of a lot in a hurry, okay? All right, so reinforcing what we're gonna talk about is, okay, again, with the all or none property of action potentials. So an action potential in a cell is the same as every other action potential once you get it going. Because in order to be an action potential, once I open the first voltage-gated sodium channel, I'm going to open all of them, right? And it's going to be really similar to all other action potentials in that same cell. So whereas, again, graded potentials can be strong or weak, action potentials are the same within a cell. So there's no strong or weak action potentials. So when you hit threshold or you exceed threshold, you get the same action potential. So let's look at that for just a second. So here's the graded potentials, right? Weak one, strong one, depolarizing, both of them. These are graded potentials. They will be action potentials once you hit thresholds because you're opening what kind of channels here? voltage-gated sodium channels. And notice that a barely threshold versus a supra-threshold, right? So let's say this is 100 molecules versus 180 molecules of acetylcholine. They get the same action potential, right? You get the same one, right? So this is the all or none nature of action potentials. All action potentials are the same in a cell once you get them going. So if the stimulus is below threshold, subthresholds, you don't get one. But a threshold or a supra-threshold one, both will get you one, right? The only difference between like how getting to threshold, like if you were slightly hyperpolarized, you still, if you get to threshold, you get an action potential, right? You still get one, right? It's just harder to get there because you're starting from a different point, okay? So the all or none nature of action potentials says that basically once generated, every action potential in a cell is the same as every other action potential in the cell, right? They're consistent with one another. Which, of course, brings about a question. You know, of course, your nervous system can tell intensity, right? You can. But how the hell do you tell intensity if every action potential is like every other action potential? So how would you tell the difference between just enough acetylcholine to start an action potential? You know, let's say 50 molecules of acetylcholine get you an action potential. Is there any difference between 50 molecules and 500 molecules? There is, but not per action potential. The difference is that a stronger stimulus gets you not bigger action potentials, but more frequent action potentials, assuming that the refractory period is working and you repolarize quickly, right? So we really depend on this. So let me give you a metaphor for it, and it kind of works like this, okay? So what is the difference between the action potentials from poking my finger with a pin, pain, right? Action potentials going in and being interpreted as pain, versus cutting my damn finger off, right? There is a difference, right? We can tell intensities, yes? So what's the difference? Let's hold it steady for a single neuron responding to that. A single, because of course there's more when you cut your damn finger off, but for one neuron. So put my finger with a pin, here's the difference. The action potentials go pain, 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 right? All at the same place interpreted in the pain in your cerebral cortex. And then cutting my damn finger off will go pain, 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 as fast as it possibly can, right? It's called frequency coding. It's how the nervous system communicates intensity. The difference between smelling something slightly versus smelling something massively is just the frequency of action potentials that are going in for the same stimulus, right? It also works on the way out. On the way out, the way that it works is, you guys haven't learned about motor units yet, have you? Okay, so I've got a single neuron that's up here in the cervical region of my spinal cord that's going to control maybe 150 skeletal muscle fibers simultaneously, right? But skeletal muscle fibers can bend minimally or maximally or somewhere in between, yeah? Okay, so how do I tell them how much I want them to bend? I either send infrequent stimuli and I'll get a little bit of contraction, middle stimuli, or really frequent stimuli to control the degree of muscle contraction, right? So it's all about frequency. Does that make sense? Because every action potential is the same in a cell as every other action potential. The way that you communicate intensity is with frequency. So the way that that would work over here is like, if we go back to this, if we say, okay, I'm going to dump 10,000 molecules of acetylcholine onto this cell body. Obviously, that's going to start one, right? But I've got so much left that as soon as it's repolarized, I'm going to start another one, and another one, and another one, and another one, right? So much stimulus that assuming that you are repolarizing properly, yes, you're going to start the next one then as soon as you possibly can, and you're out of the absolute refractory period, right? You'll send them as frequently as you possibly can. Okay, so that's called frequency coding, sometimes called sensory coding. And don't let this picture confuse you. This is just simplifying. This is a single action potential just represented as a spike. This is more frequent, and this is really frequent, right? So a little bit of threshold, subthreshold, you don't get anything. Barely threshold, you get one. Supra-threshold, but not maximally, you get more frequent. And then go crazy with it, and you'll get really frequent. As frequent as the tissue can send it, right? Does that make sense? And that depends on how quickly they repolarize. Nervous tissue repolarizes as quickly as possible. Okay, so now, interactive physiology, for those of you who use it, it's really, really good for this chapter. Okay, now, what we need to get to now is myelinated versus unmyelinated axons, because we haven't actually talked about that, right? Myelinated versus unmyelinated axons. So you guys have learned about myelin, of course, before. So everything that I've told you thus far about axons was in an unmyelinated axon. Unmyelinated axons are a bunch of little bitty dominoes that you hit the first one and they all roll over, right? And what they have is lined up all along the axon, they are going to have voltage-gated sodium channels and their two friends, right? Voltage-gated potassium and the other one. Everybody okay with that? Okay. So that works really well if the two neurons that are communicating, for instance, are right next to one another, like in my cerebral cortex. In my cerebral cortex, there are neurons communicating with neurons, but they're microscopically separated from one another. So myelination is not actually necessary there, but I have a neuron up here that is trying to control action down here. What I'm going to have to do is I'm going to have to insulate that axon, and I'm going to have to speed it up, and the way that I do that is with myelin, okay? So it's the same kind of story, but stick with me to understand how it works. So you guys learned about myelination and anatomy, right? So what you have is the axon hillock, still right here by Dana, is the first place that you have a whole bunch of voltage-gated channels, right? Okay. But then what I'm going to do next to that is I'm going to myelinate this particular region of the axon right here. And myelinate means I'm going to wrap it so tightly in cell membranes donated by other cells that there's going to be no possibility of diffusion in or out of the cell. So I'm really keeping any of the ions from moving right here, but then I'm going to have a node right here, and the node is going to have more voltage-gated channels, and then this one's not going to let anything move. So how does this work? How this works is, in a myelinated axon, I am not going to move the ions in and out all along the thing, I'm going to move them at the nodes. Okay, so if I hit threshold, right, then I'm going to open my first voltage-gated ion channels. There's a boatload of them here, so they're going to create a big wave of depolarization, okay? And then this wave, I'm not going to let dissipate because this is wrapped tightly. No ions in, no ions out. I make a big enough wave in the electrical nature of the fluid that it will open these. Yes? And then I hold on to it right here. Don't let it move at all. Make a big enough wave. Again, tons of voltage-gated channels at the nodes, right? Make a big enough wave that it will open these, right? And then do it again. So what they call it saltatory conduction. Saltar in Spanish means what? To jump. It means to jump, yes? So they think it looks like it's jumping, but I would argue that it's not jumping, it's diving, right? Because it's really diving. I don't know how to say dive in Spanish. How do you say dive in Spanish? Somebody tell me.
Bucia.
What?
Bucia.
Say it again.
Bucia.
Bucia? Okay.
Bucia.
Okay. Bucia. Bulsatory conduction. Divatory conduction. Yeah, because what's happening is I created a big electrical change and then I just held onto it in the intracellular fluid and then it popped back up, opened these channels, held onto it in the intracellular fluid, popped back up, right? Held onto it in the intracellular fluid, popped back up, right? So what does this do? It allows it to go for way longer distances, and it's actually faster than an unmyelinated axon, right? And it's really holding on to the charge to allow it to go down a myelinated axon all the way down here, right? And so the metaphor you could use is instead of a bunch of little dominoes, right? Once you flick the first one, it opens the next one all the way down. A fewer big dominoes, right? This one's big, right? This one is going to create a big change that if it's big enough, it will open that one, right? Which will knock that one over, right? So this is how it works in myelinated axons. It's kind of the same thing, only you get it started and then you protect it. You get it started and then you protect it, right? So that's how myelination works. Questions? No?
So it goes out of the whole axon and then pops back into the.
So it doesn't go out anywhere. What happens right here, so this would be the axon hillock. At the axon hillock, what happens is you flood the sodium in, majorly depolarizing that part, but then you don't let any ions move under the myelin, right? But it still can create a big enough wave, big enough electrical change that you can depolarize that one, right? This one, then this one, then this one, right? Over and over again.
So it stays within it just depolarizes as it goes through.
Yes, you only have to depolarize the nodes, but you depolarize the nodes dramatically enough that they will depolarize the next node, right? So it's a big electrical change at each of the nodes, and then you just hold on to it, right? You just don't let any sodium or potassium move under the myelin sheath. You just keep it from moving. That's why they've always described it to you as insulating the axon, right? Because it's holding onto it and insulating it. Do you guys remember what myelin looks like? So here's your axon, and it wraps it completely, multiple layers. Damn near impossible to diffuse through those multiple layers, basically like saran wrapping over and over and over again until it's super duper thick on the outside. So nothing really moves, right? In and out of the cell under the myelin. Everybody good? Okay, go ahead.
What kind of distances are we talking about between the?
It's still microscopic distances, because it's made by, Schwann cells or neurolymphocytes. They're still microscopic, but they're like a whole cell microscopic. So we're still talking. It's still not, you don't have to do every single little nanometer along the cell. You basically just get to skip a whole cell and then go again, and then skip a whole cell and go again. So let's apply this for just a second to a disorder that occurs. A disorder that occurs that causes demyelination of your long axons is MS. Have you guys heard of MS, multiple sclerosis? So I want you to think about this and see if you can kind of understand what happens with MS. This is an oversimplification, but what happens with MS, it's an autoimmune disorder in which your immune system makes a mistake and starts to destroy the neurolymphocytes and oligodendrocytes that are myelinating, just the cells that form the myelin. So this outside portion, and it's just a mistake. But what it's going to do is it's not really going to, for instance, impact like the cerebral cortex. It doesn't impact like your high-level processing, right? Those are unmyelinated in the first place. They didn't need it. But what they can impact, and they really do, is when you're trying to get an action potential to go a long distance, like to my digits or to here, right? Because I'm going to demyelinate. And I want you to think about whether the same axon that we just described as having voltage-gated channels here, but not here, and then here, but not here, would work effectively if I lost the myelination. So if I depolarize here and then take off all the myelin right here and allow the depolarization to dissipate right here, right? Would I be able to make sure that it actually got depolarized right here? No, I need to hold on to that big change unless I'm actually adding to it, right? So what happens when you demyelinate a long axon that requires myelin is that the cell just can't send the signal at all, because the anatomy of the cell is not set up for a bunch of little dominoes, right? It's basically like, I knocked this big one over, but this one was missing, right? It didn't actually hit it, okay? Does that make sense? So that's why you start to lose, for instance, sensory and motor, but mostly in the periphery first, where you really, really depend on myelination. Is that okay? Okay. So because they don't work. If it was a myelinated axon, it doesn't work when you yank the myelin off of it because it doesn't have voltage-gated ion channels under where the myelin should have been. Yeah? Okay. Okay. So now let us sort of reinforce this by teaching you a little bit of patho, a little patho. So when I teach patho, it's always because I think that when you break something, you can really understand how it was supposed to work, right? So I'm trying to reinforce this stuff. So you guys told me before that you understood that if you varied ion concentrations, they would be more likely to vary in the extracellular fluid than in the intracellular fluid. So for instance, starvation would deplete ions where first? Extracellular fluid, right? Or taking in excess would have too many ions first in the extracellular fluid. So what we're going to do is we're going to do that. We're going to vary ion concentrations in the extracellular fluid, and we're going to look at their impact on electrically active tissues, right? And these are actual disorders that truly occur. Okay, so the first one, by the way, read these every time with, this is the disturbance, and then the rest of this happens because of the disturbance, okay? So hypokalemia, hypo implies low what? Potassium, okay? Where? Extracellular fluid, that's the disturbance, okay? How would this occur? massively decreased intake, but we're talking starvation conditions, right? That would be really severe. But even more likely, massive loss of fluids because ions are water soluble, right? So persistent vomiting, diarrhea, diuretics, right, can really make you lose any of your ions massively. And for that reason, I don't know if you guys know this, but worldwide still, the leading killer of children worldwide is diarrheal diseases, right? So persisting vomiting, diarrhea, diuretics, hormone imbalance. The hormone you don't know yet, but I'm going to put it there because later we're going to learn it. And I won't test you over these hormones yet on this set of notes. Just want them to be there so that you can go back once we learn hormones. That hormone called aldosterone regulates potassium concentrations, also sodium concentrations. And then kidney failure. Your kidneys are supposed to regulate your ion composition, and if they fail, you can have all kinds of ion disturbances. Okay, so those are some ways that it could occur. So when it occurs, what does that mean? When you have too low potassium in the extracellular fluid, it means that what should be true is, of course, your potassium should be about 30 inside to about 1 outside, right? That's what it should be, okay? But now I'm going to lose massive amounts of potassium on the outside, and it's going to be 30 to 0.1, which is 301. Everybody with me? Okay. So now just let it sit there for a minute, dealing with that. What's going to happen to potassium if you let it sit around at rest at 300 inside to 1 outside? What's going to happen to potassium? It's going to diffuse out faster than it normally would. Okay, so let it do that for a couple of minutes. And then the cell is going to be sitting there. Instead of that normal resting membrane potential, which is -70, it's going to be sitting there at what? If potassium moves out at rest. -90, minus something further away. So it's going to be blank polarized at rest. It's going to be hyperpolarized at rest, okay? That's all well and good until, of course, you try to start an action potential in an electrically active tissue from a hyperpolarized state. Does it mean you will never get one? No, it's kind of like being in which refractory period? Not absolute. Kind of like being in relative refractory period all the time, right? Could you get to threshold? Yes. Dependably every time you meant to? Probably not, okay? So what happens is it's just sitting around, hyperpolarized. That makes it way, way harder to get to threshold and harder to get action potential. Is that logical to you guys? Okay? All right, so which body systems does this impact dramatically? Your electrical systems, of course. Electrical systems should have an action potential, and I freaking told them to, right? Have an action potential. So what are the symptoms? Fatigue, both mental and physical fatigue. Sluggish reflexes. Reflexes are supposed to respond when you tell them to, right? But also muscle weakness, right? You're completely exhausted because remember, your muscles depend on frequent action potentials causing stronger muscle strength. What if I don't get them every time? And then, importantly, causes cardiac arrhythmias, because what should happen with your heart is an action potential should cause a contraction when I tell it to. But if it doesn't do it dependably every time, what you do not get is a dependable atrial contraction, pause, ventricular contraction. Atrial contraction, pause, ventricular contraction. Doesn't necessarily mean it's going to stop your heart, but it sure predisposes you to it. Does that make sense? Okay? So it's a big, big deal for the heart to lose anything that provides rhythmicity. Okay? So that's hypokalemia. And then this one's really interesting. This one's hyperkalemia. Hyperkalemia is what level of what where? High potassium in the extracellular fluid. Okay, so let's think through this. With high potassium in the extracellular fluid, I'll come back to the causes in just a second. What it's going to do is it's going to make a concentration gradient that's different than normal. So what it should be is 30 inside to 1 outside, right? So now maybe it's 30 to 10, which is 3 to 1. You with me? Okay. And that will impact the charged nature of the cell, but even more importantly, I don't actually depend on potassium for depolarization, but I do depend on it for what? Repolarization, yes? So after I depolarize, I need to repolarize quickly, and that depends on that 30 to 1 ratio, but now it's 3 to 1, my 3 to 1 ratio. So what's going to happen is makes it difficult to quickly repolarize the cell. If you can't repolarize the cell, you can't use it again. It is useless, right? Does that make sense? It can't send another action potential. That's a big deal with the nervous system. It's an even bigger deal with the heart, isn't it? If you depolarize and contract it, and then you get stuck and you cannot repolarize, you cannot use that cardiac muscle cell again, right? It's a really big deal. So what happens in this situation is if you depolarize and you can't repolarize, then that cell is useless, okay? So symptoms, massive changes in EKG, of course. Severe muscle weakness. The severe muscle weakness can, if you live long enough, lead to paralysis. Usually not permanent, but it can lead to paralysis because I depolarized it and I can't repolarize it, so I can't use those muscle cells anymore. But even more importantly, this causes cardiac arrest because if I depolarize my cardiac muscle cells and they're stuck and I can't repolarize them, realizing that they all talk to one another, right, through gap junctions, then basically what I've done is like I've stopped my heart. So it causes cardiac arrest up to 67% of the time if left untreated. Super important for those of you guys who are going into healthcare because quite often you will give people potassium drips. You better make sure it's a person who needs a potassium drip because at the end of this set of notes, there is a news article about a nurse who killed somebody, giving them a potassium drip when they didn't need one. Go ahead.
So my dad's had like 5 heart attacks, and his EKGs always show abnormal. And his heart is larger than his chest. So is this what's causing it? Because sometimes it just feels like he's having a heart attack.
No, with that kind of problem, it's probably long-term congestive heart failure, hypertension leading to an enlarged heart. I don't know it.
He was just
but,
born with a big heart.
Oh, he was born with a big heart?
Yeah, he was just born with a big heart.
Well, the more hypertension you're dealing with, the bigger your heart gets to compensate. It's unlikely that that started out with an electrical issue, although I can't confirm that without knowing any more about it. But let's talk about this and how it's related to something related to social issues. Because this is also, let's talk about how this can occur. So it could occur with massive increased intake, but only if your kidneys aren't functioning normally, right? Because your kidneys will clear out excessive amounts of potassium if you're just ingesting it. So increased intake, but really only when coupled with kidney insufficiency. So you could eat 16 bananas in a day if you wanted, right? You'd probably yak, but even if you didn't and you had healthy kidneys, they would probably pee it out for you. But let's talk about burns. Okay, this also is a real risk of death for burn victims because if you have, let's say, third-degree burns over 60% of your body, think about how many cell membranes you would have damaged, yeah? And all of that potassium was sequestered inside your cells, and then you damage the cell membranes, and where does it go? Into the extracellular fluid, then picked up by the blood plasma and circulate. And if you're not looking for it, it can cause heart attacks, yeah? But then this brings us to lethal injections. With lethal injections, what we do is purposeful hyperkalemia. We do this specifically to kill people. And I'm from Texas, and Texas kills more people by lethal injection than the rest of the country combined. I'm not saying that with pride, I'm just saying that with truth, okay? If you kill somebody in Texas, we will probably kill you back, especially if you're a brown person, let's be honest. So let's understand how this works. So how does this work? And by the way, you do it here in Utah too, just not very often. So how does death by lethal injection work? Death by lethal injection is related to hyperkalemia, but really what happens with lethal injection is there's a triple cocktail. And the first part of the cocktail is to get the person to chill out because it's probably a little nerve-wracking. And the second part of the cocktail is a muscle relaxer. That's not for that person, that's for the people who are watching. And then the third part of the cocktail is a really high potassium intravenous solution. And so that third part of the cocktail, you give them high potassium and it will stop their heart. If done properly, it will stop their heart within a matter of seconds to minutes. Okay, that's how it actually works. So interesting stuff related to this is that of course, capital punishment is not universally accepted at all. It's not even state to state accepted, of course, right? There's an interesting podcast. I don't know if you guys have ever listened to this podcast. I linked it down here if you're interested in it. It's right here. The podcast called More Perfect. It's about the Supreme Court. And it's chasing lethal injections all around the world, the lethal injection cocktails. So what happens is, of course, the United States does more lethal injections than any other country. But we don't make all of the chemicals in that triple cocktail. We had to import them from other countries. And so an investigative reporter decided that she was going to try to figure out where the chemicals came from. So she started to research where the chemicals came from. And what she ended up finding out was some really interesting things, like, for instance, one of the lethal chemicals actually comes from the UK. Don't have death penalty, right? So she goes over there and she tells the people, do you know that this chemical that you're manufacturing and sending to the United States is used for lethal injection? They're like, well, we don't support capital punishment. We're not selling it anymore to them. And then she found another location. It was in Sweden. And she went to Sweden and said, do you know that this chemical that you're exporting to the United States is used for lethal injection? And they're like, no, we take the Hippocratic Oath. First, do no harm. We do not sell. And these chemicals, by the way, are not just for lethal injection. They also have medically approved purposes. So she chased it and put out fires all over the world. Very interesting. And then, of course, what happened in the United States is they couldn't get access to adequate levels of the chemicals for a few years, really. Then what happened after that in Oklahoma, I don't know if you guys heard about this, it's probably 6 or 7 years ago. They couldn't get the chemicals necessary for lethal injection in Oklahoma, and they had someone who was set to be executed. And so they came up with their own cocktail, and they thought it would work. And he lived for hours afterward. So this is directly related to physiology, and you of course, you have your own politics and your own voting, but it should always be based on actual facts. So some physiology for you to put in your tank. So that's what that one is used for. And again, up to 67% mortality, but they do extra high doses with lethal injection, extra high doses. And by the way, this one right here, this little article that I linked for you, this is a nurse who inadvertently killed a patient with a drug that was used for lethal injections in a person that didn't need potassium. She accidentally killed somebody with that. Okay, and then go ahead. Sure.
So if they have those, would that be where a pacemaker might come in? Because would that help? Because it sends a signal, right?
For if they had hyperkalemia?
Or no, is it the lower one? Hypo, right?
No, they just need more potassium. Yeah. So the arrhythmias, if you have an inherent arrhythmia, and I have one, I have atrial fibrillation, then it's not an ion imbalance, it's an electrical imbalance within my heart. And if it doesn't behave itself, I do need a pacemaker later. Yeah, it's not. Arrhythmias can be related to ion imbalances, but they're not all related to ion imbalances. Ion imbalances will always exacerbate them. Go.
So the guy who still lived for hours, what happened?
He ended up dying.
Was he suffering though? Did he
Yes. I mean, he was paralyzed, so you couldn't tell that he was suffering, but because they gave him a paralytic at the same time. So there was a huge lawsuit. Is what happened. That's why the Supreme Court ended up taking it up. So that podcast, by the way, this one right here is all about Supreme Court. I have been obsessed with the Supreme Court since I was 12. Totally obsessed with it. I am a Supreme Court trivia nerd. And I could not have been more happy when this podcast, all about all of the most important Supreme Court decisions, none of you will care about them except for this one. It's interesting. Okay, then 2 other disorders. The 2 other disorders are hypos. This one is hypocalcemia. Hypocalcemia. Now, I know I haven't told you anything about calcium related to this story, so I'm going to tell you how calcium is related to this story. This is an oversimplification, but every sodium channel on electrically excitable tissues, not every, most sodium channels on electrically excitable tissues use calcium as the latch. Extracellular calcium as the latch. Okay, which means that as long as calcium is there, the sodium channel should open when you tell it to and stay closed otherwise, right? You with me? Okay, it's extracellular calcium that's used as the latch. So if you get into a situation in which somebody has hypocalcemia, what is going to happen to sodium channels even when you didn't trigger them to do so? they're going to open. So if you let that sit around for a little while with hypocalcemia, your cells might be slightly blank polarized at rest. Depolarized at rest, right? Because sodium channels are open that shouldn't have been open. So what happens in that situation is it makes the cells slightly depolarized and easier to hit threshold, right? Easier to depolarize to threshold. So what could have been meant to be, for instance, a subthreshold stimulus like this one right here, with the cells sitting slightly depolarized at rest, it ends up becoming a threshold stimulus accidentally, right? So what you get is inappropriate action potentials in both nervous tissue and muscular tissue, which can lead to all different kinds of things, which can lead to spasms, muscle spasms. Nerves and muscles have spontaneous inappropriate action potentials. You can get cramps in both skeletal and smooth muscle. You can get spasms. You can get exaggerated reflexes. You can get convulsions with your skeletal and smooth muscle. And you can get tetanic contractions in serious situations. Tetanic contractions is when your muscles seize up and you can't actually relax them. Those are usually the tetanic contractions because of hormone imbalances that cause ion imbalances, not just like, I lost too much calcium today. So you have probably experienced just minor forms of this if you get super duper stressed out. Have you ever had that little thing that happens in your eye muscles that makes it go crazy and nobody else can see it? It's usually either because of calcium or potassium imbalances, and nobody else can really see what's going on, but if you drink a glass of milk and eat a banana, you'll probably be fine. It's related to the fact that some of your stress hormones, because it's usually when you're stressed out or when you're really tired, stress hormones cause some ion imbalances that aren't major or long-lasting, but they do cause ion imbalances. And we'll talk about stress physiology a little later. Okay, so what causes this hypocalcemia? Decreased intake, again, it has to be pretty massive, but lack of vitamin D, right? If you can't absorb calcium, you can get all these symptoms associated with it. And then there's two hormones. Again, I won't test you on the hormones on this set of notes. PTH and calcitonin are calcium regulatory hormones that I teach in anatomy. But if either of those is out of whack, you can have these problems. So what are some symptoms of long-term low calcium that can lead to hypocalcemia? Dry skin, psoriasis, coarse hair, and brittle nails. But that's not helpful because those are all symptoms also of living in Utah, right? Everything's just dry as a bone here in Utah, right? Anybody else move from like a really moist area? Did you see yourself age 10 years and you have to spend way more money on your hair and your skin? You're like, I was pretty by default before, and now I have to invest. Okay, so, and then the last one is hyponatremia.
I have a question.
Go ahead.
On the hypoglycemia, if it is long term and you are having continuous spasms, all those symptoms, and you have muscle relaxers and that they treat you with potassium and calcium, would you continue to get more time to start killing those cells off, or what happens?
No, it doesn't actually kill your cells off. They're fine. You're just going to have the symptoms until you figure out a way. A lot of times, if you're, do you guys know that most of us in Utah in the winter are vitamin D deficient? Yeah, almost all of us are vitamin D deficient in the winter. So if you can figure out levels, for instance, if you're actually being treated, they will test your blood plasma calcium level and your blood plasma vitamin D level, and then treat with that, usually prescription forms. But no, it's not going to like kill off the cells. These guys, you don't need calcium specifically for maintaining the homeostasis to keep them alive, but just to make them function properly. Okay, and the last one, hyponatremia. This is what level of what where? Decreased sodium, extracellular fluid. Okay, I'll come back to the causes in just a minute. Let's talk about what happens. What happens with the decreased sodium in the extracellular fluid is it screws up the concentration gradient for sodium, right? The sodium should be 15 outside to 1 inside, right? Lose most of it, and now maybe it's 5 to 1. Good, okay? So it's not as steep as it should be. So when you try to get an action potential, you can't depolarize quickly enough to get dependable action potentials because the concentration gradient is screwed up. So it can cause muscle weakness, skeletal muscle. It can cause nausea, and then it has some interesting impacts on the brain, but not directly related to this, but also really important. So muscle weakness and nausea, it's usually, by the way, associated with a lot of water fluid loss. So puking like your guts up. Have you ever gotten like really kind of dizzy and a little off from like losing tons and tons of fluid? So you're probably suffering from all of the hypos, but this one will make you feel a little bit wackadoodle because in addition to losing a whole bunch of water and a whole bunch of sodium, you are also going to screw up the tonicity of your cells. So sodium is your primary extracellular ion, right? You lose a whole bunch of it, and now your cells, instead of sitting in an isotonic solution, they're sitting in a what? Hypotonic solution, which will make them swell. Okay. Brain cells swell, they act wacky. You are going to have personality changes, you may even have hallucinations, you may have violent behavior. This one lands people in the ER left, right, and center because they are acting wacky and pissing people off. And they're going to take you in there and they'll be like, obviously on drugs. You may be on drugs, but maybe not, right? It could just be that you've got hyponatremia. But luckily, usually what comes with this is like this person is like aggressive, but they're also super low blood pressure, so even if they tried to take a swing at you, they wouldn't be able to connect, right? Okay, so how does this occur? Since this is the most common electrical imbalance. Well, it occurs far more in the summer than it does in the winter because of course, sweating, right? Decreased intake, not a problem for Americans at all. Decreased sodium intake, excessive sweating, vomiting, diarrhea, or diuretics, right, can make this occur. Kidney failure, of course. Hormone imbalance, those two hormones you'll learn later, they're water and sodium regulatory hormones. Poor diet combined with lots of diuretics like beer is a good way to get this, especially if you add summertime. Yes? So this is Texas in the summertime, pretty much. Also football, full pads, right? So football, full pads in the summertime, right? Add some beer to congratulate yourself, and then sweat your butt off the entire time, right? So the issue is, especially with alcohol, is that alcohol is a diuretic, yeah? It's a fluid that you're taking in that you're going to have to pee out, but then you always pee out more than you took in because it's a diuretic. Especially problematic with Utah beer, right? Utah beer is a little better than it used to be, but it's a diuretic that has lots and lots of water in it compared to other beer. You're way better off doing tequila shots, I promise. Okay, so what's going to happen is you are going to lose tons and tons of water and the sodium that goes with it. And it can cause these ionic imbalances that make you act like a wackadoodle. If you're also drunk, it's a little hard to figure out why you're acting like a wackadoodle, right? So you just go to the hospital and then they shoot you up with some stuff and they're like, oh, they're fine, okay? But so, and then the other one that is kind of famous for this is ecstasy or MDMA use. So we will talk about what ecstasy actually does to your neurophysiology a little bit later, but ecstasy, it has killed very few people, far fewer people than alcohol, for instance. And I don't know if you guys know this, but it's in a lot of clinical trials for PTSD and that kind of stuff right now. But I'm talking about off-label MDMA use, right? Like I'm going to go to Burning Man, I'm going to do MDMA, and it's in the middle of freaking summer, right? So it's hot as hell. You're going to take some MDMA. It depresses your thirst response, but it makes you able to dance in the desert for 8 hours straight. Can you see how this can and does, in people, lead to hyponatremia? Yes? Okay. So you end up in the hospital with hyponatremia, and you're usually going to live through it, right? But that's how it actually does. So we shouldn't die from youthful indiscretions. Otherwise, every interesting person you would ever know has been dead. Yes? So what you should tell your experimental friend, who is definitely not you, for sure, is to take some damn Gatorade, right? Because you need to replenish both your fluids and your electrolytes. Otherwise, nobody's ever going to hear that amazing story that you have to tell later, right? That might have a tinge of regret, but not when you're talking to your best friend. It doesn't. Not at all. Okay, so that is hyponatremia. So let me just show you a couple things. Last little tidbit is this one will allow you to practice. I linked it on the bottom right here, okay? And this is a place where you can practice looking at action potential. So just watch for just a second. That is threshold, and then you can watch things move, but that's too far away. So let's look at the cell a little closer up. And then we can make it go fast forward. We can make it go slow. We can make it show us the charges, the concentrations, the ion chart, and then we can go again. So you can see what's happening, and this is just something that you can play with. For those of you who are very visual and kinesthetic and really need to see what's going on, you can also slow it down all the way. You can make it go fast forward, you can stimulate it, right? And then watch what happens on the graph. So that can help reinforce for you guys if you want to practice on that. Okay, so then we are going to, with our last 15 minutes, start the endocrine system. Go ahead.
Well, I was just looking at the chart. So when would the refractory, like the absolute refractory period and the absolute refractory period be?
The absolute refractory period is from here down to about here, absolute. And then from here and then into the hyperpolarization phase is the relative. So relative is always right after absolute, right? Relative is after all the sodium channels have reset, but your potassium channels are either still open or you're hyperpolarized because they were open, right? So it's usually right about here.
And absolute, so there's no way you can.
No way, because you haven't reset your dominoes, so you can't start another action potential. Everybody okay? Okay. All right, so we're not going to do anything hard on the endocrine system today, and the endocrine system is on the second test, but I do want to get you started thinking about it because you do have a lab about it this week, okay? So with the endocrine system, so first off, let's sort of address the fact that most of you guys did not cover the endocrine system in anatomy, and that is simply because it's not really an anatomical system. So really all it is a bunch of different secretory cells in a bunch of different places that communicate with one another via the bloodstream, but they don't look anatomically like they're a body system, like for instance the nervous system does, right? You got the brain, you got the spinal cord, and it's all connected via nerves to the rest of the nervous system, right? Or cardiovascular system, I got a whole bunch of tubes and pump, right? Digestive system. So the pituitary gland controls my ovarian secretion, but they don't have anything to do with one another other than the bloodstream, which literally everything has to do with one another. So it's kind of pointless to teach you the anatomy of the endocrine system. We'll just throw it in when we learn physiology. Because what we have is a bunch of different secretory cells in a bunch of different places that are actually communicating with one another. So the endocrine system, by the way, for those of you who use interactive physiology, it's fantastic for the endocrine system. The endocrine system is really one of the body's two major control and communication systems. It is releasing hormones to communicate with other cells, primarily for what kind of communication? Intrinsic or extrinsic? Primarily for extrinsic. And then the other one that helps with the endocrine system is the one that does the same thing but more quickly, and that's the what system? Nervous, right? So those two are your extrinsic long-distance communication systems. Good. And mostly they're trying to maintain homeostasis. Okay, so endocrine organs, by the way, are not all glands. I know we're used to talking about endocrine glands, but I will tell you that although the pituitary gland is a gland and secretes a hormone, the heart secretes a hormone too, and it's definitely not glandular tissue. So I try when I remember to say endocrine organs instead of endocrine glands. So what they are going to do is they are going to secrete a hormone, which by definition we know travels through the bloodstream to find its target tissue or target cell. And a target cell is any cell with a blank for the hormone. Receptor, right? A receptor that's specific to that hormone. Everybody good? Okay, so once it gets there, what does it do? Just to give you some idea about the complexity of the endocrine system. So the hormone gets dumped from the secretory cell. It actually, by the way, gets dumped into the interstitial fluid, but typically those tissues are really vascular, so it gets picked up immediately into the bloodstream. So it gets in the bloodstream, and then it goes, let's say, from the secretory cell to the target cell, okay? And it can go straight there without like anything particularly happening to it. And that's that could occur. There are other things that can occur, and we'll come back to that in just a second. So let's say it gets straight from the secretory cell to the target cell without anything happening to it on the way. What we still have to do after that is it has to bind to the target cell receptor, right? And then, so I am growth hormone, and she is the receptor, and all of you guys are the organelles within her, right? Okay. So what has to happen, for instance, with growth hormone, is that I have to bind to her. I have to stay outside, though, right? And she has to initiate, again, I know you hate the term, but a signal transduction pathway, right? That will result in all the way back there, Carla, who is the nucleus doing protein synthesis. Does that make sense? Okay, so there's a bunch of crap that has to happen in the cell, right, after it binds, and that's called, again, the signal transduction pathway. Okay, so assuming that we went the simple way, then you have the stimulus and you have the response. But note to self, because you have hormones and they might go bad on you, it's more complicated than that quite often. I don't test on the more complicated things. I just want you to know that it could occur. So sometimes it doesn't go straight from point A to point B. Sometimes some stuff happens to the hormone when it's in the bloodstream. One of the things that can happen is some hormones are released in an inactive form, and they have to be activated before they will cause a response in their target cell. Testosterone is one of those. Testosterone is made from the testes in an inactive form, and it will not cause a target cell response unless it's activated. Why the hell does that matter? You can have a male who looks like he has all the symptoms of low levels of testosterone, but you test his testosterone levels and they're totally cool because it wasn't activated. Yes? And why does it matter? Well, an endocrinologist does not treat those two disorders in the same way. He doesn't need more testosterone, right? He needs to activate the testosterone that he has. Good? Okay. Second, what looks like a hormone problem could actually be that the hormone is getting broken down too much. Like if your liver or your kidneys are overactive, that's one of the ways that you break down hormones. So I could be releasing, for instance, totally adequate levels of thyroid hormone, but my liver is malfunctioning and breaking it all down. And that's not a thyroid hormone disorder, although it looks like one and would have the symptoms of one, right? It's actually a liver disorder, okay? So you could over-metabolize the thing, or sometimes hormones actually have to work with one another. And so what looks like a disorder with one is actually a disorder with another. So let me give you an example. Thyroid hormones are metabolic hormones, meaning they support the proper metabolism in all of your cells. Good? Okay? And growth hormone primarily causes protein synthesis and cell division. And that should make sense, right? Growth, protein synthesis, and cell division. But does it make sense to you that your body won't commit to making growth hormone if it doesn't have adequate levels of thyroid hormone? Yeah? So sometimes the two hormones actually interact with one another. So why do I tell you this? To make you cry and think the endocrine system is really, really complicated? It totally is. It's also understandable in the way that we learn it. But just so, if you are dealing with an endocrine disorder, you need to see an endocrinologist. Because this shit is hard, right? And you do not learn it deeply enough in the first years of medical school. You probably need to specialize, right? You need someone who realizes that a single hormone can have impacts all over the body, and that what might look like a secretion problem is actually a liver problem or a target cell problem, right? And it's really complicated. Super interesting. Endocrinology, if you decide to go to med school, you are never going to be poor if you're an endocrinologist, because there are only like 7 or 8 of them. Maybe Shelby knows. Do you know how many there are? Endocrinologists in Salt Lake? Last time I checked, there were under 10, but there may be 20 now, but that's still very few, because how many doctors are there in this area? Right, pediatric endocrinologist, or an endocrinologist that specializes in diabetes, or an endocrinologist that specializes in reproductive endocrinology. So, and sometimes you can be on a waiting list for 6 months before you actually get an appointment with one. So,
6-7 years of post-medical school training.
Yeah, it is crazy the amount of training. It's hard stuff, man. Okay, so let's talk before you guys go, because you're going to have lab over endocrinology, or you already had it this week. Let's talk just a little bit about what happens with endocrinology. So I want to talk about basically what happens with your hormones is, since hormones tend to regulate lots and lots of functionality in the body, you do not, for instance, release a solid, stable amount of hormone. You release how much insulin you need versus how much glucagon you need. You release how much estrogen, and too little is bad, and too much is bad. So the primary way that you are going to regulate these levels is with negative feedback loops, right? So that you can maintain homeostasis in the hormone levels. But there are really 3 different things that, 3 different ways to actually regulate and feedback. So let me show you each of the 3 of them, okay? So sometimes what happens is a hormone is regulating a nutrient or an ion, and it's the nutrient or ion that feeds back to tell you when you don't need any more hormone. Sometimes it's a hormone that feeds back and tells you when you don't need any more. and sometimes it's the nervous system. So I want you to look at A, B, and C, and as I draw these examples, you do not have to memorize these examples yet. I just want to draw real ones so that I don't confuse you for later. Try to see these examples, how they fit into A, B, or C by looking at, again, what feeds back. So one that we already know, right? And this is oversimplified, but high blood glucose is detected by the pancreas, which releases insulin, and insulin goes to target cells, a bunch of them, and decreases blood glucose. So, and what feeds back? What feeds back is the new level of blood glucose, and it feeds back to the pancreas to say, that's cool, we don't need any more insulin yet. So does that fit into A, B, or C? Is it the hormone feeding back? Is it a non-hormone substance like a nutrient or an ion, or is it the nervous system? Non-hormone substance, right? Good, okay. All right, so let me give you another example. Don't worry about the details of this one, just look at the bottom and the feedback mechanism so I can show you what we're talking about here. Okay, so this is an example. The hypothalamus, we'll learn this later, secretes a hormone called TRH, which goes to the anterior pituitary, which causes it to secrete a hormone called TSH, which goes to the thyroid and causes it to secrete two hormones, T3 and T4. These are hormones. And in addition to going to their target tissues, how do you know when you have enough of this? The T3 and T4 feed back both to here and to here. So what example is this? What's feeding back? Hormone. So that is B. Yeah? Okay? Okay? Either way, it's accomplishing the same thing. It's just not a nutrient or an ion. And then the last one is hard to draw, but it's easy for me to tell you the story about it. Okay? You can draw it if you want to, but here's an example that Tiger walks into the room. My nervous system says, hey, I know Tiger's big and scary. Yes? Oh crap, Tiger. Yes? And what it does is my nervous system tells my adrenal medulla to secrete epinephrine or adrenaline into my bloodstream. Okay? And the epinephrine or adrenaline does what it does. Okay? But then what happens is I leave the room and the tiger is behind, right? I'm totally safe. And then my nervous system says, hey, no more tiger, right? Stop secreting adrenaline. So what is it that started it and what is it that stopped it? Was it the hormone coming back to my brain to say I've got plenty of adrenaline, or was it, it's the nervous system, right? Nervous system turned it on, change in nervous system stimuli, turned it off. Another good example, you want one more? Oxytocin release that occurs during nursing, right? So the stimulus of suckling on the nipple will actually cause milk letdown, right? And the milk letdown does whatever it does, and then the suckling stops. And then what stops the oxytocin release? Just the lack of suckling, right? So that was all sensory and nervous system related, okay? So that is where we will stop today. This week you're doing endocrine in lab, and then next week you're doing muscle. They're going to get ahead of me for a few times because we have a test next week. I'll catch up with them as soon as I can.
So how's the format? Is it like choice written?
It's the same you were used to in anatomy.